Next up, we have Spencer Karau of the University of Ottawa. He is discussing key functional plant traits to predict the rate of colonization of invasive plants in North America. Thank you for joining us, Spencer.
Yeah, it's a pleasure to be here. Can everyone hear me just fine? And can everyone see my presentation?
Yeah, perfect. Your presentation looks good to me.
Great. All right, I'll jump right into it. So hello, everyone. My name is Spencer. I'm a master's student at the University of Ottawa, and today I wanted to talk to you a bit about the research I'm doing for my thesis. So my thesis is entitled Using Biological Traits to Predict the Rate of Colonization of Invasive Plants in North America. So why is this topic something that is important? Well, we have a lot of research in the literature that studies the impacts of invasive plants on the ranges that they invade. And there's also a lot of research that looks at what factors really go into facilitating the rate that these plants spread. But what we don't have is sort of an agreement on which factors are the most important. And there's really a lack of clarity on what in particular facilitates spread. Additionally, there's no reason to suggest that invasions aren't something that's going to keep occurring. And what's more is that with factors like climate change, it's possible that invasions are going to be occurring in places where in the past they never would have had the chance to occur. So getting a better understanding of how these plants spread in novel ranges and what actually facilitates the spread is a good idea for helping managers when they have to deal with these invasive plants. So what's the question we set out to answer? We wanted to know which, if any, biological traits can be used to predict the rate at which invasive plant species will colonize newly invaded ranges. So to answer this question, we need to sort of set up a variety of moving parts. So I'm going to try and give a bit of a background on everything, and then we'll jump into the results. So the first part we should take a look at is, what are biological traits? When we talk about biological traits, we're talking about the intrinsic characteristics of the plants themselves. These are basically the features of the plant that make the plant the way it is, or the species. And we want to know how these traits relate to spread rate. So for our study, we looked at 3 traits. The first trait we looked at was seed dry mass. Typically, plants with a heavier seed are more resistant to tough conditions, and they start off with more resources. So this can make them more tolerant of unfavorable conditions. But species with heavier seeds are also typically harder to disperse. So with that in mind, we have sort of 2 hypotheses, and we're going to make 2 hypotheses about each of our traits, and we'll explain what we mean by the terms in the hypotheses a bit later. But for now, we predicted that species with a higher average seed mass will reach climate equilibrium faster, but will have lower maximum colonization rates. The second trait we looked at was plant height, just how tall is the plant at its maximum height. And we thought that, or it's known in literature that plants that are taller typically can outcompete those that are shorter for light and other resources. So with that in mind, we thought that plants with a higher maximum height will reach climate equilibrium faster, but typically takes longer to grow larger. So we hypothesized that we predicted that plants that that are taller will have lower maximum colonization rates. And finally, we looked at specific leaf area. This is just the ratio of the leaf's area to its mass for a species. And typically high specific leaf areas are related to high relative growth rates. And with that in mind, we predicted that species with high specific leaf areas will reach climate equilibrium faster and have higher maximum colonization rates. Okay, so we're talking a lot about spread, but how do invasive plants usually spread? Well, they typically follow what's known as an invasion curve, and this is just a way to look at how these species propagate once they arrive in a novel range, and it can be broken down into several stages. The first thing that happens is transport, whether it's brought over on the bottom of a boat or brought over for ornamental purposes and escapes. A species somehow makes its way from its native range to North America. After transport, a colonization period begins where the species has to establish itself in the novel range and build a self-sustaining population. Once this has occurred, the plant can begin spreading across the landscape. And this is typically where the maximum rate of colonization is observed. And finally, after the plant has had time to spread and it's sort of reached the extent of where it's going to be in the novel range, we say that the plant has sort of reached equilibrium. It's no longer increasing its distribution and it's no longer decreasing its distribution. So typically when we think about invasion curves, we think about them in geographic space, but we can also think about them in climate space. And this is important for us because when we look at the rate of colonization of our species in our analysis, we look at it in both geographic space and climate space. So quickly, what's the difference between geographic space and climate space? Well, geographic space is simply put, you can think of it as where plants or species are found on a map. So in this example, you could say that the species is in the northeast quadrant of the map, or the plants are in 25% of the range. And if these plants were to spread across this map, fortunately, the geographic space would increase as well. Meanwhile, climate space can be thought of as what environmental conditions are the plants found in. So if you considered each of these different colored squares on our map to be a different climate based on how warm and how wet it was, we could split this range up into 5 different climate spaces. And if we see that our plant is in 2 of those 5 climate spaces, we could say the plants occupy 40% of the available climate space. So with that being in mind, we can see how even though the plants are in the same spots in reality, the amount of geographic space and the amount of climate space they inhabit can be different. In addition to traits, we also included some other factors in our analysis. First factor was native range size. We included this in models that were dealing with expansion of species in geographic space, and it's just how big was the plant's range in its native range. The second factor we included was growth form. This is just a categorical variable. Is the species a tree? Is the species herbaceous? Is it a climber? Is it a shrub? And finally, we also had native niche breadth. This is just a measure of what sort of climates or niches was the species found in its native range. Was it found in a broad range of niches, or was it found in very specialized niches? And we use this covariate in our models that we're dealing with expansion in climate space. Another thing to note is that when we ran models that used plant height, we actually removed the growth form term. And this was because plant height and growth form were seen to be significantly correlated with one another. So where is all this data coming from? Well, our current data is coming from the Global Biodiversity Information Facility. Our climate data comes from WorldClim, and our trait data is coming from TRI, the global database of plant traits. These are all excellent open source databases that are curated and have data submitted to them all the time. And without the generous submissions of researchers, this type of analysis wouldn't be possible. So for my study, we looked at 258 species that were identified by the Center for Agriculture and Biodiversity to be invasive to North America. These species came from a variety of different native ranges and were from a variety of growth forms, albeit the majority of which were herbaceous. So I collected and cleaned data for all 258 of these species, and I was able to find data for 247 of them. And of these 247, 105 of them reached equilibrium in climate space. Okay, so we have our biological traits that we want to look at. We have our other factors in our analysis. And we begin answering the question of, did traits influence colonization rate? So the first response variable we're going to use to answer this question is time to equilibrium. This is just the amount of years that it took for a species to reach equilibrium in climate space since its introduction in North America. So if a plant was introduced in 1850 and it took until 1950 to reach climate equilibrium, time to equilibrium would be 100 years. And we calculated this for every species that reached equilibrium in climate space. And you can see that on the y-axis of our plots here, that the times to equilibriums for our species range from 25 years to 125 years. On the x-axis, you can see our traits, and note that these x-axes have been logged. And what you'll see is that there's a lot of scatter here. Our points aren't really conforming to the line of best fit. And it doesn't seem like there's a great correlation between our response variable and our trait variables. What I'm showing you here are the individual terms that go into each of these models. So for example, our top model, the model would be comprised of seed mass, a native niche breadth, and growth form. And when we put these terms together to make a prediction about how long it took a species to reach equilibrium in climate space, so the time to equilibrium, we can see that the adjusted R-squared for that model is quite low. And it's quite low for all of our models dealing with time to equilibrium. What's more, we found that adding the trait terms to our model actually did not improve the predictive power of the model in all cases except for with plant height. So That is to say, when we added seed dry mass or specific leaf area to our model with our other covariates, the predictive power did not measurably change. Okay, so the second response variable we used to try to answer our question was looking at rate of spread, specifically the maximum rate of spread in climate space over a 30-year period. So to calculate this response variable, we plotted our occupied climate space over time at 5-year intervals, and we did this for every species. We calculated every possible 30-year slope, and we isolated for the highest slope. And doing that, we were able to get a value for every species. And we wanted to look at the data in terms of rates because only 105 of our species were able to reach climate equilibrium. So by looking at rates, we were able to leverage all 247 species into our analysis. Doing this, you'll see our response variable once again on the Y and our trait variables on the X, and note again that the traits have been logged. We see that there is again a good deal of scatter, and it doesn't seem like our trait variables are well correlated with our response variable. This is echoed in that the adjusted R-squared values for our models are again quite low. And what we noticed here was that actually none of our trait values serve to increase the predictive power of our models. So adding the traits in did not improve the predictive power of our models in any of these cases. So finally, we had one more response variable that we were going to look at. This was looking at the same thing as our second response variable, but rather looking at spread rate in climate space, we were looking at spread rate in geographic space. So it's the exact same calculation as before, but looking at things in terms of geographic space. And what we found was that when we had our final set of response variables, we had to log it to normalize our data. We wanted to look at the data in geographic space for two main reasons. Firstly, most of the current literature is focused on expansion in geographic space. So by doing this, we can more readily compare our results to what's out there in the literature. And secondly, the sort of barriers to expansion might be different in geographic space and climate space, and likely are different. And some traits might prove to be more important to spread in one space over the other. So by looking at things in both geographic space and climate space, we can start to look into that. Doing this, we get probably our best correlations between our response variables and our trait variables, although they are still not very strongly correlated. And note again that the trait axes have been logged. And what we'll see is that while our adjusted R-squared values for these models are highest that we have seen, they are still quite low. Note that because we're dealing with geographic space here, our native niche breadth term has been replaced with a native range size term. And similarly to what we saw with time to equilibrium, the only trait that actually improves or makes a measurable difference on the predictive power of our models is plant height. Adding seed dry mass or specific leaf area to their respective models did not serve to increase the predictive power. So what does this mean? What can we say? What can we garner from these figures? Well, of the three traits we looked at, the only one that really seemed to make an impact on predictive power of our response variable was plant height. And it was only able to do that for 2 of our 3 response variables. Moreover, when it did make an impact, the overall adjusted R-squared value of the model was still quite low, with the highest one being less than 0.2. Meanwhile, our other factors, our covariates in our analysis, seem to be significant predictors the majority of the time, but as we saw from our adjusted R-squared values, the overall predictive power of these terms was still quite low. We also noticed that in most instances, adding the trait terms to our models did not seem to increase or decrease or make a measurable impact on the predictive power of our models. So this suggests that whatever limited predictive power we are getting is coming from these non-trait factors, from these covariates. So what can we garner from all of this information? Well, it seems that the traits that we looked at, our study traits, do not well predict spread rates at large scales or in general contexts. So when we calculated our species averages, we considered species from research that was performed in their native range and in the invaded range. And we aggregated all that data to create species averages. And doing that, it seems like it was hard to sort of see any correlation between a trait and a response variable that was tied to colonization rate. The other factors of our analysis, like native range size and native niche breadth, seem to play a greater role, but as we saw with our adjusted R² values, these roles still seem to be relatively limited. So all this, along with information in the literature, seems to suggest that perhaps at smaller scales, or when we have a higher data resolution and we know more about the specific context of the environment that's going to be invaded, we could say more about the predictive power of a trait, or a certain biological trait might be more predictive. But when we look at things at this wide scale and we are sort of grouping these varied landscapes together, it's really difficult for us to say anything about the predictive power of traits. So thank you very much for your attention, everyone. This work would not have been possible without the following people, and I'm really happy to take any questions.
Thank you so much, Spencer, for your presentation. We have a few minutes for questions, so if anyone could direct your questions to the Q&A box, and we can answer from there. While we're waiting for Spencer, I'm just wondering, I'm sure a lot of viewers here understand what it is, but it's kind of basic question. Would you be able to explain to me like what climate equilibrium means exactly? That's probably a very broad question, but
Yeah, it's a tough distinction to make, and it's one that we sort of, it's hard to explain. So when we say climate equilibrium, we're basically talking about the point in time, or the point at which a species has encountered all of its all the climate spaces that it's going to encounter. So if you look at this example, you can see that this range has divided into 5 climates. So when a species arrives, it's going to enter into the first climate space that it's going to encounter. And as it spreads, it's going to encounter more and more climate spaces. So over time, the species will continue spreading and continue encountering different climate spaces, but eventually it's going to stop spreading, and it's not going to increase the amount of climate spaces it enters, and it's not going to decrease the amount of climate spaces it enters, theoretically. And that's the period of equilibrium. So it's not approaching more climate space, and it's not retracting from climate space. So the amount of climate space that it occupies is at equilibrium.
Perfect. Thank you. That makes sense. Thank you for that
Yeah.
explanation, Spencer. And we do have a question in the chat. If you were to do additional analysis to answer the question to predict which species will be invasive in the future, what variables might you investigate further?
So that's a good question. We're kind of limited in the variables that we could select, in that we are dealing with open access data. So we have to sort of choose from what we have. So let me just read the question.
It would be in the answered box. Yeah.
So I'd really like to do some more analysis on traits related to, species. Yeah, so variables. So I'd really like to do some more analysis into variables related to dispersion. So sort of what dispersal method are these plants using, and how far do they typically disperse? And I think that would be a good trait to look at, and would speak a lot to the rate at which a plant spreads, and also the time that it takes for them to reach climate equilibrium.
Thank you, Spencer. So there's time is up, but there is one more question. Wouldn't reproductive traits be more predictive? I'm wondering if you could answer that in the Q&A box of chat. And also you can leave your email if you want to for any further questions.
Sounds good.
Thank you so much, Spencer. That's super interesting.
Thank you very much.
Thank you. Okay.